Hi guys, let us today talk about a new topic called metagenomics. Metagenomics came into the field of biology a couple of decades ago, maybe during 2000 or 2001, 2002, during that period, when genomic studies were going on, when Human Genome Project was also going on. Then suddenly this metagenomic approach that evolved in the field of research. What actually it means, metagenomics, it deals with the whole set of genomes. In an environmental sample, you can say. Means in an environmental sample, there are large number of microorganisms that are thriving there. But when you try to culture them, you end up with merely 1% of the total community present in that environment. So 99% of them are not growing in your media. Whatever media you provide, you don't know exactly the nutritional requirement of those microorganisms. So the problem arises how to study them. The approach that is taken is called culture independent approach. Culture independent approach. This approach it studies those organisms which are basically viable but not culturable. Means they are present in the system, they are living, they are doing their job. In fact, they might be the most predominant one in the particular habitat, but particular habitat in terms of their niche, in terms of their function, but they are not culturable yet. It is better to say yet, because in future you might culture them by designing certain media. So you have to take a culture independent approach to study them. There are approaches like microscopic study, or the one of the biggest approach that has arisen is the metagenomics study. So here comes the metagenomics. What it does, it analyzes the total set of DNA that is present in an environmental sample, and automatically it will involve all the organisms, all the DNA of all the organisms, whatever present in that environmental sample. So it will take into account even those ones which are not culturable and the one which are culturable also. So if you analyze the DNA of an environmental sample, you are analyzing the genomes of those organisms, and you end up with analyzing the microbial community in that particular environment. What kind of environment we are talking here? It may be any environmental sample, like it may be agricultural, field soil, it may be forest flow, it may be animal gut, or it may be human oral microorganisms. So you can take samples from anywhere and you can subject to this metagenomic studies. What actually is done in metagenomic study? First of all, you have to isolate the DNA course with which you are going to handle, and then in today's world, since the gene sequencing has become so easy in the world of new generation sequencing technologies that have come up, and the cost is also very reduced, one goes for the total genome sequencing, whole genome sequencing of the environmental sample. The total DNA that has been obtained is subjected to sequencing. So whatever sequence you are getting of this A, T, G, C, and etc., now you start analyzing these sequences, the variability present in the sequences. It has been seen that the variability in the sequence is 1,000-fold more than what is present, what was expected, or what was previously studied about the microorganisms, because people did not know that what they are studying in the culture is merely 1% of the whole. Anyway, so this is the way how metagenomics can be done, but there are simpler ways to do it also. You can go for PCR of with this the total DNA that you have isolated. PCR of what? PCR of 16S rDNA sequences. So you have to design primers for 16S rDNA, and you can subject this DNA to PCR amplification. The DNA that will come up will be the 16S rDNA of all the genomes present in that DNA sample, in that environment. So if you analyze if you go for sequencing this 16S rDNA, you may end up with a number of sequences that are the true representative of the number of organisms present, number of species, in fact, not organisms, number of species present in that particular environmental community. So this is one way. Another way is you can go for restriction analysis of this 16S rDNA. And this has been called RDRA, amplified rDNA restriction analysis. But of course, I should mention here that RDRA is suitable for cultured organisms. That means those ones which have appeared in the culture. You can identify them using this RDRA technique. But doing RDRA with a DNA sample isolated from the soil or the environment, whatever you are talking about, doing an RDRA will be futile, I think. So better after doing a PCR of 16S rDNA, you can subject it for a kind of gel electrophoresis that has been called DGGE, which is denaturing gradient gel electrophoresis. Means it's a gradient of gel of polyacrylamide, where the concentration of which decreases from downward to upward, and it is prepared with the help of a density gradient maker. So if you load DNA over here, 16S rDNA that has been amplified by PCR, and since the sequence this PCR, this 16S rDNA is basically a mixture of all 16S rDNA sequences that are present in this DNA sample, which has been purified from an environmental sample. So it's in fact this DNA contains sequence of all the organisms present in that environmental sample. So here they will be separated in a denaturing density gradient gel. They will be separated because it is run at 60 degrees centigrade and it is a denaturing one. The minor differences that exist between the 16S rDNA will make them separate in the gel, and that can be picturized and that can be counted, and number of bands is representative of the number of species present in the environmental sample that in question. So this is one way of analyzing the microbial community. Another way what you can do is you can do RISA. What happened? RISA. RISA is what? RISA is ribosomal intergenic spacer analysis. Means in the genome, in the DNA, say this is a 16S rDNA gene and this is 23S rDNA gene. So there is a spacer region between these two genes. This is called intergenic spacer, ribosomal intergenic spacer region. If you design primers from here and here, and if you amplify this portion, this is variable. This portion will be will not be similar in all the organisms present there in the environmental sample. So the DNA that represents the total environmental sample will have different sized RISA, and if you run a gel like a denaturing gel of this kind, the bands will be separated, and those bands will be representative of species, a particular species. So RISA is a very good technique by which one can identify or analyze organisms present in the sample. If you are doing a whole genome sequencing, you have to go for bioinformatics research further, and you have to do what is called data mining, which is a difficult job, but it can be done using different types of programs like R programming, or there are tools available for doing that, bioinformatic tools. So this is all about metagenomics. In fact, if you are doing a 16S RDN analysis by sequencing, this has been given a new term called metatranscriptomic. Because you are analyzing the transcript, that is 16S RNA. From you start it with the RNA by doing RT-PCR. You amplify that and you go for sequencing. You can go for sequencing the whole set of mRNAs to do a metatranscriptomic study to get a better idea of the total community present in the system. These are the things related with metagenomics. I think it will help you in your further studies. If there is any error, please let me know, because this is a new technique, newly arisen horizon where you can play around with your DNA, and there may be different other methods of doing that. One can also, other than metagenomics, there are approaches of studying these culture-independent microorganisms. One is called PLFA, that is phospholipid fatty acid analysis. This also tells you about the predominance of particular fatty acid, tells you about the type of organism present in that environment at a particular time and particular interval. So it helps to study the ecological success in a particular environment. The technique that I was telling about, the microscopic technique, that is also called FISH. FISH means fluorescent in situ hybridization. You hybridize the DNA or the cell with stents like DAPI, and you see the microorganisms under the microscope. But the problem is their shape, which are not very variable, and most of them have this kind of shapes, by which you can say you can go up to a limited extent the type of organisms present there, but each of them, a spherical may be of different type of species. A rod may be represented by different types, so quite a number of species. So by microscopic study, it is not very good way to understand the microbial community in a system, but it also exists parallelly with these metagenomes. Thank you very much for your silent listening. Thank you.